Hi, so my name's Sam Virtue, and I work here at the Institute of Metabolic Sciences. And today I'm going to be speaking to you about the concept of metabolic flexibility. It's a little bit of an esoteric concept, so I'm going to start by introducing it and then go on to try and explain what we think it can tell us about metabolic dysfunction and metabolic disease. So, to understand metabolic flexibility, you first need to understand how our bodies handle nutrients. And I think that the first things we're going to talk about, the first point is going to be pretty obvious. How do nutrients get into us? Well, they get into us through our mouths. We eat food, and that food can be healthy or it can be quite a bit less healthy. But the key point is if we eat more nutrients than we expend the energy that we've consumed, we will gain weight and we will become fatter and ultimately potentially end up obese. The second point, which is how do the nutrients we have accumulated get out of us, may actually be a little bit less obvious. Because actually the nutrients get out of us, not say, for example, in feces, but through really two routes, one of which predominates. So those routes are in our breath, where we can lose carbon dioxide and water, and in our urine, where we can lose water and we can lose urea, which is how we get rid of nitrogenous waste, and nitrogen comes from protein. But the really critical route for losing weight, if we want to lose adipose tissue, fat mass, is we're going to lose it through our breath, because the carbon backbones that make up fatty acids can only be lost in the form of carbon dioxide from our bodies. So we literally, if we want to lose weight, we need to breathe away our adipose tissue. Now, I've shown these two slides, and in the first one we had on the left-hand side, we had all the food shown as food, as what we would eat, pizzas or donuts or tomatoes. But when we were showing how we were getting rid of it, we were showing it as molecules, as carbon dioxide, as water, as urea. But we can actually think about what we eat in terms of chemical formulae as well. And they're actually a lot more simple than you might think, given we eat a complex diet. Basically, what we eat boils down to three types of molecule. The first, amino acids, which we're not going to talk about too much in this talk in the interest of simplicity, they're made up of carbon, oxygen, hydrogen, and some nitrogen, and that can only be removed in nitrogen and urea. But we also eat carbohydrates, symbolized by bread here, and they are a carbon with a water or hydrate. And finally, we also eat lipids. These are our major nutrient sources, and that is a carbon bonded to two hydrogens. So these usually come in long chains called fatty acids, but this is the basic unit. And if we want to convert lipid or carbohydrate into carbon dioxide and water and expel it in our breath, what you can see is we're going to need something else. We're going to need oxygen, because if we look at this, we have two hydrogens and one carbon, and we need to turn it into a CO2 and an H2O. So we're going to need quite a bit of oxygen to fully eliminate this nutrient. Carbohydrate, we need a bit less oxygen because carbohydrate comes with some oxygen in it. And we can actually use the amount of oxygen we need to produce our CO2 and water to get some information about what our bodies are using. So to explain this more formally, we can use a technique called indirect calorimetry, and this is normally thought of as a device for calculating energy expenditure from oxygen consumption and carbon dioxide production. But because of this lack of ratio, this lack of a one-to-one ratio between CO2 and oxygen, the fact it varies depending on whether you're burning fat or you're burning carbohydrates, means that we can get information about the nutrients being used, and we generate a ratio called the respiratory quotient or the respiratory exchange ratio. They mean the same thing, which is the CO2 divided by the O2. And so just to show these equations formally, RQ is VCO2 divided by VO2. And burning carbohydrate has an RQ or an RER of 1 because here is a unit of carbohydrate. To fully convert that to carbon dioxide and water, we require 1 molecule of O2, 2 atoms of oxygen. And so therefore, we need 1 molecule of oxygen, for 1 molecule of CO2, a ratio of 1. Lipid, on the other hand, this has an RQ of 0.7, because in order to fully oxidize this, a unit of lipid, which has a hydrogen, and a carbon, we're going to need 1.5 molecules of molecular oxygen in order to get 1 molecule of CO2. So 1 divided by 1.5 comes out as about 0.7 in practice, because all of these fatty acids that we're burning have a carboxyl group on one end, and that has a bit of oxygen in it. So it's not quite the perfect mathematical 0.66. Okay. So we could sort of think that if we have a fixed ratio of carbohydrate and lipid in our diet, then we could calculate how much carbon dioxide and oxygen we would need by adding up how much of this that we have and how much of this we have, and working out how much oxygen we need to turn it into this. However, our bodies are somewhat more sophisticated than that, and we've evolved complicated storage mechanisms. So what do we do after a meal when we are healthy? Well, we consume a mixture of this carbohydrate and lipid. And the lipids, if we are healthy, we'll store them in our adipose tissue and lock them up until later. The carbohydrates we will burn, and some of the carbohydrates will also be stored acutely in the liver as glycogen. And finally, if we're in real excess of carbohydrate, we can even turn some of those carbohydrates into lipid by the process of de novo lipogenesis for long-term storage. But the result of this is, even though we've eaten a mixed meal, we are burning almost exclusively carbs, and our RER or RQ will be 1, pure carbohydrate oxidation. Now, when we're asleep, we should somewhat reverse this process. So what we should do is we should take some of this lipid we stored earlier in our meal, and we should release it from our adipose tissue. And then that will go to be burnt. But not every cell or organ in the body can run on pure lipid. Some of them need carbohydrate. And so our liver will release some of these carbs, this glycogen we've stored, in order to fuel red blood cells in particular and the brain. And as a result, the RER we will have at night can get down to 0.8, and if we're fasted even longer, get down even lower to maybe 0.74, something like that as an absolute minimum. So in other words, even though we're eating food which may have a sort of varied composition, we have systems to control what gets burnt when. So what is metabolic flexibility? Well, metabolic flexibility is describing how well our bodies can do those processes of storage and those processes of release. And it's the ability to make the transition between using the carbohydrate in the day after we've eaten to using the lipid at night, and vice versa. And in order to assess it, we use a calorimetric technique. And this study is from David Kelly. It's been done in humans. And in this case, they're doing calorimetry not on a whole person, but across a leg. And the way that it's done is you put cannula into the artery, cannula into the vein, and you measure how much carbon dioxide and oxygen go into the leg and how much come out. So in this state, they've got a pseudo-fed state, which is a clamp. So this is a technique where you pump insulin into someone, and then you measure their blood glucose, and you pump glucose in order to prevent them from becoming hyperglycemic and dying. And the more glucose you need to pump in for the same amount of insulin, the more insulin sensitive you are. So in lean, healthy individuals shown in these white circles, then when you pump insulin and glucose into them, their legs become almost entirely reliant on carbohydrate. And they use that glucose, which is a carbohydrate, and they oxidize it for fuel. Conversely, in the fasted state, these lean, healthy individuals, their adipose tissue is releasing lipid, and they're able to get down to an RER, in this case, of about 0.8384, and they're using the lipids that are being provided to them. And this difference between the fasted and, in this case, the pseudo-fed state, delta RER, can be considered metabolic flexibility. What we can see when we look at the black squares, the obese individuals, is in the fed or pseudo-fed state, the clamp state, these individuals do not reach as high an RER. And this is because they will be obese, they will have some insulin resistance, and their muscles will not respond to the insulin to take up the glucose properly. And as a result, they can't burn it. And so they will have a mixed nutrient usage of lipids and carbs. Interestingly, in the fasted state, we see a higher RER in the obese individuals. And we're going to come on in a minute to explain why that is the case. But the overall result is, instead of having a nice sharp sloped line like the healthy individuals, these people have an almost completely flat RER profile between fed and fasted and are inflexible. They cannot change the nutrients they burn dependent on their nutritional status. So at the heart of understanding metabolic flexibility for us is adipose tissue. And there are two forms of it. One of them is called brown adipose tissue. I'm not going to talk about today. It's a thermogenic organ that converts energy to heat. The other form is white adipose tissue, and this is the body's main energy store, and it has three main functions. The first two, long-term nutrient storage, which can be months in humans, and also as an endocrine organ, are very well known. The one I've started introducing to you today has really only been looked at for maybe the past 20 years, and it's this role as a lipid buffer that I've spoken about. And the fact that after a meal, it can take up fatty acids and store them and release them again when needed. And we really focus on this third function because we think it's really important for understanding metabolic disease and also its ability to then control metabolic flexibility. And analogous to the leg data we showed, adipose tissue can also become metabolically inflexible in obesity. So this is another AV difference experiment, but in this case, instead of being across a leg, it's across a fat pad. And instead of measuring carbon dioxide and oxygen, we're measuring free fatty acids, the components of the lipid we eat in our diet, and we're looking whether they're taken up or released. Now, in a lean individual, each time they eat a meal, the adipose tissue has a net uptake. It stores fat, just as we would expect from our cartoons earlier. And this makes sense because this process of storage is insulin sensitive. And as we can see, each meal, the adipose tissue, the fat tissue, continues to take up lipid. And then at the night, when we go to sleep and start our fasted, the adipose tissue releases fatty acids to fuel our body. In the obese individuals, you can see that the insulin-sensitive uptake of fatty acids is blunted. They're not as good at doing it. But then in the fasted state, the obese adipose tissue is also worse at releasing it. And this makes sense because if you have an obese person, if their adipose tissue is bad at taking up lipid but can still release it, they'll very soon lose their adipose tissue and cease to be obese. And what you can realize then, looking at this, is if the adipose tissue isn't releasing fat, then there's no fat for the muscle to burn. And that explains why those obese individuals in the fasted state were more reliant on carbohydrate, because they're not getting the lipid delivered to their legs. So to summarize what we can think about in this case, what happens after a meal when adipose tissue is now unhealthy? Well, we've taken in the same nutrients as we did before, but now we're going to block the storage of lipid into the adipose tissue, and potentially block this process, the storage of the newly synthesized fat into adipose tissue. As a result of this, instead of this lipid being locked away and saved, it's going to go and get burnt. In addition, we're not going to be able to burn everything. We're also going to see that lipid accumulate in organs like liver and muscle. And when you accumulate lipid in organs like liver and muscle, it poisons them and it causes insulin resistance. And the result of this is because we're now burning a mixed meal, we're going to have an RER of maybe 0.84, 0.85, 9, 0.9, something like that, because we're not doing what we should and storing our lipid. Okay, now I'm a murine physiologist, so our interest was, could we study this in mice? Because then we could look at the genes regulating it. And the problem is these AV difference experiments I've shown you in humans, you can't do in mice. The mice are too small. So how do we go about it? Well, in humans, we do the AV difference experiments because humans are annoying and eat different foods. And over time, if you're constantly eating lots of carbs, for example, your RER profile will tend to wibble up. So your daily circadian cycle of RER will get higher on average. Equally, if you eat lots of lipid, this will drive down your daily circadian cycle of RER. And this means it's difficult to get good assessments from free living respiratory exchange ratio. Mice, however, though, conveniently, we keep them in cages and we feed them the same food every single day. And as a result of this, there is a fixed ratio of carbon and oxygen going into the mouse, and this means there'll be a fixed ratio of CO2 to oxygen. And this ratio can be called the food quotient, the theoretical amount of CO2 that will be produced to oxygen consumed for any given food. And so one thing we can then see is, do mice have an average RER over 24 hours that's the same as the food quotient? Well, one of the problems is you have to account for the fact mice essentially have a very large capacity to change their body weight. They can change it by 10% in 24 hours. So, and if they're losing weight, they'll be burning their fat reserves and that will drive their RER down. Whereas if they're storing it, they'll be burning more carbs like our humans and their RER will go up. So we need to correct for the delta body weight. So if we regress our change in body weight during our calorimetry run for our mice against our respiratory exchange ratio, we can see what the intercept would be if the mice were weight stable for 24 hours. And indeed, we can see it pretty much is the same as the food quotient. The food quotient of this diet is 0.91, and our regression cuts the y-axis here at 0.9, so almost the same. And that's within the margin of error for our data. But the mice can still do the storage. So they can fluctuate their RER because they still have this system, the capacities to store their lipid and burn their carbs. And in fact, they're even better at it than humans. And as a result of that, we get a really nice circadian profile of RER. It's reversed from humans, but what you can see here is during the light phase when mice are asleep, you get lower RERs. And then during the dark phase when they're eating, you can get RERs that even exceed 1. Because that process of turning carbs to lipids has an RER of 5, and that can drive the RER above 1. So in this system, we've been talking a lot about the ability of fat to store lipid and release lipid and how that can control the nutrients we use. What happens if we have no fat? Well, this experiment has been done, and there was a mouse model made called the AZIP mouse, and that has no adipose tissue at all. So this is just showing adipose tissue here. And what you can see when we look at the RER profile, in this case over 2 days, is we can see the nice wild type mice in red doing a really lovely profile of a curve in RER, using lots of carbs, storing their lipid, using almost exclusively lipid. The AZIP mice, and this is just data from 2 mice, but it's so strong the effect, you can see it super clearly, they are almost completely flat. They are doing the hypothetical thing of burning everything they eat as they eat it. And this has important consequences because in the absence of fat, you just have the RER equaling the diet. You also get some insulin resistance when you have overload the system, you have too much lipid going at once, then you're going to have that stored in liver and you're going to get insulin resistance. And this is exactly what we saw when we tested this. So this was some work we did in collaboration with David Savage's lab, who works here at the IMS on lipodystrophy. You can see here 4 days of calorimetry. The first 2 days, the mice are on a low-fat diet. The second 2 days, they're going to be on a high-fat diet. And in black, you can see the nice wild-type mice doing a nice circadian rhythm in terms of their respiratory exchange ratio, whereas you can see the AZIP mice running along nice and flat. Now, when we switch to the high-fat diet, which has a food quotient, instead of it being about 0.9, it's about 0.78, what happens is the wild-type mice just carry on with the same average RER. They don't really drop their RER in response to it, because what they do is they store the fat and they carry on just burning the carbs. So if the diet is 60% fat, they're storing the 60% and burning the remaining 40% carbs and amino acids. The AZIP mice with no capacity to store, their RER collapses down almost immediately to be the same as the food quotient of the diet. And so that happens super, super fast, and they don't gain the body weight. But they're not gaining the body weight, but they've got all this lipid and all this excess nutrients coming in. And what that does very rapidly is causes insulin resistance. And we can just see here, blood glucose, we have the 3 groups here are the wild type as on low fat and high fat diet, and the AZIPs on a low fat diet. And then we switch them to that high fat diet for 2 days. They become profoundly hyperglycemic, full-blown diabetes, and they're also insulin resistant. The larger these bars are, the more insulin resistant they are. So that's an extreme example, but most obese people have lots of fat. It just doesn't work well. So can we model a milder form of this, where we have a bit of a problem with lipid release and a bit of a problem with uptake? And what does that look like in our mice? And this is where we get to why we think metabolic flexibility is important. So to do this, we used a mouse lacking a gene called PPR gamma, and in specifically PPR gamma 2 as a regulator of metabolic flexibility. And PPR gamma is a key transcription factor in adipose tissue. It's necessary for forming adipose tissue. If you don't have it at all, you can't make adipocytes. But it also regulates mature adipocyte function, and it controls the genes necessary for lipolysis and lipid uptake. And if you get rid of it, you have less lipolysis, less uptake, and your fat becomes inert. And it has 2 isoforms. They differ on the protein level, but the key thing for us was that gamma 1 is expressed widely, and the second isoform, gamma 2, is expressed almost exclusively in adipose tissue. So when we deleted this gene, we could just look at the effect of reducing PPAR gamma in adipose tissue. And the first thing we saw in these mice was that lack of PPAR gamma 2 did make the animals metabolically inflexible. It's not as extreme as the AZIP mice, which were a flat line, but they had a compression in the amplitude of their respiratory exchange ratio. Now, the interesting thing about this is, in the morning, these mice were over-reliant on glucose. Their RERs were too high. They were above the black wild-type dots. And so what we did to test whether this was truly glucose utilization or something else was going on was we gave them a bolus of glucose at this time in the morning when they were appearing to be over-reliant on glucose. And not perhaps unsurprisingly is that the PPAR gamma-2 knockouts, which are crying out for glucose, they're using too much when they should be using lipid, they actually cleared that glucose bolus faster. So this is a glucose tolerance test. In the wild types, it goes up to about 17. In the gamma-2s, it went up to 13. And so these mice had better glucose clearance. Now, this is a subtly important finding. And the reason for that is, how do we define diabetes? Well, we define diabetes based on this test, this glucose tolerance test. And you are defined as diabetic if you have a larger glucose tolerance test. So formerly, our gamma-2 mice, based on definitions of diabetes and prediabetes, were healthier than our wild types. And that goes against everything I've just told you, because I've been saying adipose tissue and good adipose tissue function is important for health, and obesity is a state of bad adipose tissue function. But it's not quite the whole picture, because we also looked at lipid metabolism in these mice. And what we saw was, at night, the gamma-2 mice had a lower RER, which was indicative of the fact they were using too much lipid. And this was what we saw in the cartoons with the bad adipose tissue function. In the fed state, the lipid's not being locked away in the adipose tissue, it's going off to be burnt. And so could we challenge the storage capacity? And the answer was we could. We gave them a gavage of olive oil. And when we gave them that gavage of olive oil, they were spectacularly impaired in their lipid clearance. So whilst their glucose metabolism appeared a bit better, their lipid metabolism, the gamma-2 shown in white, they were much worse at taking up and storing it. And this is important because we think that lipid handling is an early stage event in metabolic disease. So we looked at these mice when they were older, and we did one of these clamp tests. And what we found was that the glucose infusion rate, so the amount of glucose we had to pump into our gamma-2 knockout mice in order to get their glucose back to normal in response to a fixed dose of insulin, was about half of a wild type. So they were de facto insulin resistant. And so this was at 12 months. All the data had shown us at 3 months. So the key point is we think, whilst their glucose tolerance may be better or okay at 3 months, these early stage events of failed adipose tissue and failed metabolic flexibility or impaired lipid handling, and that then goes on to manifest in later life as impaired insulin sensitivity. Okay, so I'm going to give you some overall conclusions here. Metabolic flexibility describes the ability of organisms to switch from glucose to lipids as fuels for energy. After eating, healthy organisms store their lipid and use their carbs. When sleeping, they switch to use the lipids released by the adipose tissue. When adipose tissue stops working, lipids don't get stored or released properly. They end up in liver or muscle, causing insulin resistance, or they get burnt. So the defective storage causes too much lipid to be burnt after a meal and leads to lipids accumulating in other organs. And the defective release means the organism uses too much glucose when asleep and fasting. And points 4 and 5 together manifest as metabolic flexibility. And given our data from the gamma-2 and a couple of other models, we think the metabolic inflexibility, if we can measure it and intervene to target it, it's important because it occurs before clinical signs of metabolic diseases like diabetes and the test as a GTT are observable. So it's an early stage event. And it also occurs before, and I haven't shown you this data, before insulin resistance can be detected. So we know with metabolic diseases, if we can intervene early, the better it is for the outcome of the patient. Okay, so it just remains now for me to thank a few people, in particular, my boss, Antonio Vidal-Puig, whose lab I work in, and also our funders, the British Heart Foundation and the MLC and the Wellcome Trust. So thank you very much.